So the topic of my presentation is meta-pangenomics and other tongue twisters. First, I would like you to understand why I chose this topic for today's presentation. I'm trying to answer this big question: what constitutes good health? For this, there are a variable number of factors. Now, for example, Agatha Shimanek here would say maybe it's Polish genetics. Maybe I would say no, it's the way I live in India. Maybe Piotr over there would say that it's the diet that he has, being such a fit gym person. Most of us now, especially with the previous presentation, now we've seen the effect of bacteria and the health. The combination of all of these factors with the bacteria that live in and around us is called the field of metapangenomics. Just to be able to analyze all this information together, I would like to present to you, like step by step, how we can build to get to this stage. So first, an overview of myself. I am a computer science engineer from India. I have a minor degree in microbial technology. Right after my bachelor degree, I was able to apply for a master's in Belgium, where I achieved this thesis where I created a database for the pan-genome of Pseudomonas aeruginosa. Now, this bacteria is quite ubiquitous in nature, and it is notorious for causing these secondary infections in immunocompromised individuals. This is a good example as well because in your gut profile, you still have traces of this bacteria, but it can cause so many problems if the different conditions are present within the patient. So just to explain what pangenomes are, in simple terms, we are essentially looking at multiple genomic isolates or strains of a given species and collecting the information of what makes them conserved with reference to each other and what makes them unique. This information can basically be achieved by extracting the coding domain sequences and then creating these persistent shell or cloud groups. So this is actually directly from my thesis, where we studied like 47 genomic isolates, and you see this U shape in the number of gene clusters. So essentially there's a large percentage of conserved genes among all the strains of the species. Makes sense. But then also there's a lot of variability if the more genomes you include. In fact, we've also seen that the more number of genomic isolates you start to include in your pangenome research, you start to decrease your core cluster. Here it's a simple dendrogram and like a cluster map of presence absence of certain gene clusters, and you see like high number of like persistent genes in the first segment. Then there's like a bit of a transition zone where it may or may not be present, and then you have like extremely unique for certain isolates. Now, how can this observation of pangenomes really help us? There is this very recently concluded research on the human pangenome, where up until now we had more of an American or Eurocentric view on how the human genome really looks like. In this research, they essentially included a large number of human genome references from Asia and Africa. I believe like there's a larger concentration of them. They used this pangenome as a reference in read mapping. So when you want to like reconstitute your genome assemblies, and there was actually an 8 times higher coverage in the mapping of these VNTR regions. These are basically the variable number tandem repeats. In a human genome, there's like very repetitive regions, and sometimes because of inaccurate assemblies, we might miss them. Using very accurate pangenome, we can have a more accurate assembly. Looking at bacteria, this is a good example. Someone was even talking about Streptococcus pneumoniae in the beginning of this presentation. There was a very interesting research where researchers looked at the pangenome of this species, and they were able to conclude that certain genes that were earlier considered essential for the survival of the species were actually quite strain dependent, and there were certain genomic isolates from these environments that were containing these accessory genes that were more essential for their survival. This is like a big impact from looking at the pangenomes. Coming back to me, I am currently a bioinformatician at Perseus Biomics. It's the same company as Agatha S, and we are currently doing microbial metagenomics using optical mapping technology. This is basically helping us achieve sequencing-free mapping of DNA. I will show you a very interesting slide in just a few seconds. Currently, our focus is on the human gut biome. How is metagenomics done for current research? We basically begin with a complex community sample. It might be a fecal sample, sample, soil sample, a water sample, and then we try to extract all the microbial DNA present in there. For us, the focus is on bacteria, so that is mainly the DNA we focus on here at the library preparation stage. I think some of you might be aware of these 16S and shotgun metagenome sequencing technologies, where the DNA can be prepared in either of two ways. Either we look at the ancestral ribosomal gene in order to amplify it and then create the DNA library, or we can have an unbiased fragmentation of the microbial DNA and then we prepare the library. After this information is created, we sequence everything and use computational tools to reconstitute them into the proper assemblies. There are several problems with this whole process which can create to bias in our final analysis or interpretation of the results. I've listed three here. First is that we are using amplification during the DNA library preparation. If certain species in your metagenomic sample is over amplified, it can actually lead to under or over representation of different species, because finally you are looking at the abundance profile of your sample. It can lead to different kinds of conclusions which may not be accurate. Second is that we are using very small fragments of DNA. I think when we talk about reads, currently we have 150 base pairs to 300 base pairs, roughly around this, to create our assemblies. And finally, as I also mentioned, the computational tools required here may be very high. So if you want to include a large number of samples, it is not very easy to scale up. What's the solution? Perseus Biomics. We're basically using optical mapping technology in order to counter these limitations with the current benchmarks. How does optical mapping work? Essentially, we begin with extraction of the metagenomic DNA, which we do not amplify. We are fluorocoding them with certain DNA motifs across every bacterial species, and then we stretch them on a polymer surface. Once these DNA stretches have been prepared, we present them to a microscope and take images. Afterwards, there are the introduction of image processing algorithms which can extract the DNA fragments and assign them to taxonomy records. What does this process in practice look like? Basically, this is the extracted DNA that is going over the surface, and then you see these linearly stretched DNA traces. We can literally see bacterial DNA on the polymer surface. The image processing algorithms that we've developed can extract these singular DNA traces and assign them to the proper taxonomy records. Here, for example, E. coli fragment, which is 54 kilobase pairs long. Compare this to the 100 and 300 base pairs reads that we were using in the traditional metagenomics processes. There is a huge improvement. Now, how is this actually comparing in turnaround time, other computation resources? Quite well. With optical mapping, the whole process from extracting the sample to seeing the taxonomy profiles takes 3 hours, because we are looking at essentially straight long reads. We can do a strain level resolution. Currently, I think with 16S, we can go down to genus level, and then with shotgun sequencing, we can go down to the species level. Here, we can go even further to the strain level. Since we are not doing amplification, there are no associated costs related to the buffers or any other library preparation or any such tool. This is simple analysis that I did actually. So we compared the profiles that we detect with optical mapping to shotgun and the 16S. It's simply a benchmark study to see how we compare. And at the phyla level, so these are basically the phylum that we detected on the first, and you see the relative abundance of each phyla. There is a great correspondence between all of the benchmarks, but if we want to go further down, we can even see species level resolution for optical mapping and shotgun. Here, it's a bit naive of me to say this right now, but in certain cases, I even noticed Bacteroides cellulositis, I suppose it's this one report, like the 7th one. There is a higher detection in shotgun, but in the optical mapping stage, we detected a higher Bacteroides fragilis, which I believe is more accurate one because we had longer reads to work with. We actually assign a better accurate kind of taxonomy. On the lower left side, you actually see the correspondence, the correlation of shotgun results results with optical mapping, and they are going as high as 0.8. This is a very nice study that we did with a human condition study. Essentially, we fed them a diet over a week. You can see this is for the same person over 1 week. The microbial profile can actually fluctuate quite a bit. Without diving quite deep into these results specifically, I would just like to say that for a single individual, the microbial profile can indeed be vastly different at different time points. What is the end game of this kind of research? As we are moving more towards higher sanitized world, there have been research studies saying that the usage of broad spectrum antibiotics are causing more and more children to have allergies and even fall sick, have asthma cases. So we really need to understand how are the bacterial interactions that we are removing by usage of antibiotics affecting us. The first question to ask here is what bacteria is present right now? And this is where Perseus is coming. Once we have this information all nicely loaded, we can welcome a field called metapangenomics. If we assume that we have, for the same individual, the microbial profile at different time points, we can start to use the concepts of pangenomics and start to create these core, shell, and cloud, basically accessory clusters. Once we know conserved regions are, we can start to investigate why this variation occurred over time. In this hypothesis that I mentioned here, CDSG, which was present in the first time point, has mutated to CDSG1, and now it's possibly performing the same functions as the first time point functional. Basically, it's causing functional redundancy because it is creating replacement for the absence of these genes in the second place. Now, this might seem like a hypothesis, but this is how current research is going. As a very quick example, there's this consortium in France right now of 45 institutes that have come together to create a meta-pangenome of human fecal matter. I suppose it's over 6 months. They want to study the effect of antibiotics on their gut. Their main goal is to study this, which species are the fastest to recolonize and which ones have the resistance to them. There is also this very interesting research I found. This is a recently concluded research study on the oral cavity of 30 individuals. They created a meta-pangenome of 3 species of the oral cavity, and they were able to prove that for all individuals, if you have dental plaque, it's either one of these 3 species which causes essentially the highest abundance in the dental plaque, because they are basically producing these bacteriocin compounds which are like chemical warfare against each other. Either one of these will always succeed. With research like this, we are actually able to see the evolution and functional dynamics between strains in complex sample. Eventually, this will also help us demystify the 2 kilograms of microbes present within us. That is where I see the future of metagenomes also lie. That brings me towards the end of my presentation. Essentially, I think bioinformatics is a very easy to integrate field. You can branch out into new topics. For me, for example, combining my thesis topic to now where I'm working, it was interesting to see how different ideas like this can combine and make a completely new domain. I would like to say that if you are suffering from decision paralysis, like choosing where and what fields to go into for your future, search for tools that you are already familiar with, try to see how the application of these tools has evolved over time. I wanted to suggest this small point to try to rebuild small research projects on your own from existing research papers. A lot of this data is public, so it is quite possible to do so. And also to keep on familiarizing yourself with new terminology and technology using these bioinformatic reviews. I have recently mostly seen them on, but they can be a bit biased because the authors can write about tools that are more favorable to use according to them, but it's still interesting to see their point of view. I think people who are looking for a very high volatile, passionate jobs, they can look for startups. I found this nice link, the public funding space. This website collects the information in Europe of the recently publicly funded startups. If you are interested, you can directly contact them and then have a better communication. I don't know if you've also walked through LinkedIn and seen the number of jobs that are temporary, they don't actually follow through. And also keep on contact with your professors and your friends and seniors. And my final note here is that most of the information that I showed here in this presentation was from the last 10 years. So there is only more new terminology and more new research that can go on from here. So cheers to making more tongue twisters. Thank you.